Real pleasure to welcome everyone to the 23rd Annual Jeannette and Bernard S. Post Memorial Lecture. Dr. Germano is going to introduce Dr. Henderson at the end of my beginning intro here, so I'll save that for her. I want to thank the members of the lecture committee who scour the best and brightest, the entire country for the best and brightest. Cal, Peter, Isabel, Chris, for finding and then coordinating for Jaimie to come here. Dr. Post, who, as you all know, was the chair and is now chair emeritus. He established this lectureship, among many other things, for the memory of his dad, Bernard, and his mom, Jeannette. She was really a woman ahead of her time, a litigator and one of the only women in her field at the time. Cal has fostered a spirit of collaboration and innovation, research, and the list of previous lecturers you can see really manifest that. Used to be that we would focus on tumor orientation, but you can see now that we have a very, very broad spectrum of people who have been honored as the Post lecturer. Because Jaimie has not spent much time at Sinai, I want to just give a little bit of a background of Sinai. And for those of you who've seen this your 100th time, this will be your 101st time to see it. It's a relatively old institution, beginning in the 1850s. And there have been 8 chairs so far, starting with Dr. Ellsberg back in 1914. I was lucky enough to come after Cal, and here you can see one of the early pictures of us together. It was a very daunting task to step into Cal's shoes, and I'm still trying to fit into them. When I started, and actually going back to when Cal started, before he came, there were 4 faculty and 400 cases. During his chairmanship, he grew that to more than 2,000 cases and more than doubled, even tripled the size of the department. One of the most important lessons I learned from Cal, I'm not sure why this is on automatic, it shouldn't be. Is it possible to change that, to take it off of automatic? One of the most important lessons I learned was how Cal interacted with the previous chair, Leonard I. Malice, in fostering their relationship. And let me just do it a different way. That way I don't have to worry. He fostered the relationship with Dr. Malice, and Dr. Malice worked with us for a number of years. We all sat in the back, and Cal and Dr. Malice would sit in the front of our conferences, and we learned as the 2 giants would discuss the cases of the department. Some of the early people in the department, Dr. Sen and Noel Perrin, were really the backbone when I joined. And Dr. Germano joined. Here you can see your program director in one of his younger photographs, and Kevin Yao, who was here doing a wonderful case just a couple of days ago. Cal always made it part of our experience to involve his family. And here you see Linda. I'm sorry she couldn't be here today. So he taught us a lot about family and neurosurgery. As you can see, an early picture of Dr. Germano and I. This is at Alana's wedding, Cal. So I won't spend too much more time on that, but I just wanted to thank you once again, Cal, for everything you've done, and especially for this lecture. Fast forward to today, the department is a big department, and here you can see our faculty and residents. The surgical volume has grown over the years, as I like to show, and you can see now we're in the 6,000 case range. We look at Manhattan discharge data, and not just me, but everyone looks at it. And here you can see that for Manhattan, Mount Sinai really has the lion's share of neurosurgery, defined as here for brain surgery, everything other than spine surgery. Yeah. Because I think, that may be a problem for anyone.
No problem. You start me. So we do not want that. And it should be good now.
Thank you so much.
Nope.
The department has been looked at in other ways. This is an article in JNS from August of 2023, looking at training programs. And by faculty size, you could see that Sinai is number 3, and by volume, number 7 nationwide, at least by the data, the way they used and collected data in those days. Since this is a talk on neuromodulation, if you will, I guess you could say that BCI fits into that category. I just wanted to highlight some of our faculty who are very, very interested in neuromodulation, sometimes called functional neurosurgery. This is a group that is focused on movement disorders, psychiatry disorders, epilepsy, and pain, with some fairly major contributions to the literature. And one of the largest experiences, for example, with RNS in Dr. Gatton's work. Brian is a very strong individual in this area. And here you can see one of his interesting studies. He's going to be the principal investigator of the Transcend study, which should be an exciting extension of psychiatric applications. Talking about BCI, there are a number of important companies out there that deal with direct cortical BCI. And we are fortunate enough to have 2 of the founders of these 2 important companies, Precision with Ben Rappaport and Synchron with Tom. And here you can see the generalized concepts. Today we're going to hear a little bit about a slightly more invasive, if you will, or more intrinsic method. And I'm very excited to hear what Jaimie has to say. I was telling Jaimie a little bit about biodesign, and hopefully he has a chance to visit with Ben a little later. This is the MedTech device incubator for our department, with a bunch of engineers, very talented people and students. We did loosely model it after Stanford biodesign, although with our focus here being primarily on device development, as opposed to things like drug development and process development and software. We're really focused on devices. And then we've had a number of successes over the past few years. One of the things that I think is really developing nicely is our technology readiness level paradigm for bringing technology from concept to company. And here you can see the way we are viewing our these are some of the various projects in the pipeline that we update every quarter. And here, these are the 4 that are most advanced now, ranging from an ICH cooling mechanism to a new valve mechanism for endovascular procedures, ENT, and cardiac as well. We are going to have a symposium. Unfortunately, looks like Jaimie is busy that day, but hopefully we will have a lot of participation. Right now, there's quite a bit of excitement about our BCI symposium coming up in November. So with that, I will turn it back over to Isabel, who will complete the introduction for Jaimie. Thank you.
Good morning. Thank you, Dr. Post, and thank you, Mrs. Post, for securing this lecture. It's been a pleasure over the years having these incredible speakers. And thank you to Peter for arranging this. Peter, Dr. Morgenstern, and Maggie and the staff in the office put a lot of effort into this. Jaimie, last year when we spoke, I couldn't believe that actually it would take over a year to get you here, but I'm so honored and thrilled that you're here. Jaimie and I have known each other professionally for about 20 years or so, done multiple programs, projects together, and it's been a pleasure to keep track of one another on the two different coasts. By the way of introduction, Dr. Henderson did his undergrad at WashU and then went to medical school at Rush, where he also did his residency, and then, as you see on the slide, had fellowships at Loma Linda, as well as in Portland, Oregon. It's very difficult to summarize in one slide, and so I apologize for leaving out so many things that Dr. Henderson has been fundamental and crucial in the field of neurosurgery, neuromodulation. As you can see here, he is a neurosurgeon, he's a scientist, and he is also professor of neurology and neurological science at Stanford University, and he has been there for the past, I would say 20 years or so. He specializes in functional neurosurgery, epilepsy, pain, movement disorder, and deep brain stimulation. He has worked extensively in neural interfacing and translational research. He was involved in the first pilot trials of DBS for tremor in the US. He has implanted well over 700 DBS since 1997. And his practice is still focused on clinical and advancing the science on neuromodulation. In the laboratory on brain-computer interface, he has really done a lot of fantastic things, and hopefully can share some of those with us today. He is the principal investigator of the BrainGate 2 clinical trial, and he has implanted sensor arrays in clinical research participant. He has a fantastic neural prosthetic translational laboratory with Krishna Shenoy, and he has been advancing the delivery of clinical viable brain-machine interfaces for individuals with paralysis. And hopefully he will touch upon some of these great latest accomplishments. So with that said, please join me in welcoming Dr. Jaimie Henderson. It's very tricky, it moves by itself.
Okay. Well, I think we'll have to switch over to my computer now. So if we can do that, should hopefully be straightforward. So thank you, Isabel. Thank you, Joshua, for the wonderful introductions. It's a huge honor for me to be here to give the CalPost lecture in 2024. It's really quite a humbling and distinguished group of neurosurgeons that I have now the privilege of being part of. So I'm going to, I think we probably ought to finish about 9 o'clock, so I probably need to keep moving things along, and hopefully I'll be able to do so. To tell you a little bit about some recent advances in brain-computer interfaces, intracortical brain-computer interfaces. I have a number of disclosures. I work with several companies that I consult with. And importantly, this is we're going to discuss today a non-approved investigational device, which is the BrainGate 2 neural interface system. So how many people know who this is? Okay, all right, so about half the room. Every year it gets less and less and less as people get older and older, or younger and younger, of course. So this is Christopher Reeve. He played Superman in the movies and was really the kind of prototype of strong and invincible nearly. And he was involved in a horseback riding accident, which left him as C1 quadriplegic, and he was unable to move anything at all. If we look at his brain, of course, he ran his foundation, he would lecture, he would do all sorts of things. So the cortex here is, of course, intact. It's really this interruption between brain and limb that we would love to be able to reestablish. The question was, until not that long ago, what happens here? Would you still be able to get signals from the part of the cortex that had been disconnected from the rest of the body? And it turns out that the answer is yes, you can. And I'll show you a little bit about quite a few things about this as we go forward through the talk. We can take those signals, we can feed them into a computer, we can do all sorts of mathematical legerdemain, and then send those signals back out once they've been analyzed as digital signals to do useful work in the outside world. And that could be anything from running a computer cursor to running a speech prosthesis or reanimating a limb, or just about really any of the functions that we could do physically. The hope is that we could restore those with this type of a bridge between the injured nervous system and the outside world. In our laboratory, we do this with a small sensor which was developed many years ago at the University of Utah called the Utah array. It's a silicon microelectrode array. This one happens to be 10 by 10, but these are usually on a 400 micron pitch. So they're very closely spaced electrodes, and they're inserted using a pneumatic insertion tool into the outer layers of the cortex. And so this is showing to scale what these electrodes look like with relationship to the neurons. And this is actually, this is how big the neuron is compared to each of these. So we can record individual action potentials from either one neuron or many from each electrode, as well as local field potentials. Route those out through a gold wire bundle, which is actually a real pain to work with, but we won't get into the surgical technique too much, to a pedestal, which is attached to the skull. So this is a titanium pedestal that allows us to transcutaneously send signals out from the brain to the outside world. And so this is what a typical, excuse me, this is what a typical spike panel looks like. And you can see on each channel, there are either one or perhaps several action potentials that can be differentiated. So if we look at the activity of any individual channel here, and this is really based on work that was originally done in non-human primates, some really foundational work done many years ago. If you ask a monkey, for example, so here's a macaque monkey doing a 8-target center-out task. So the monkey is fixating on a central point, a target appears, and then the monkey plans a move and then reaches. And if you just pick at random one of these neurons from the motor cortex, what you'll see is that they exhibit this feature called cosine tuning. So as you reach, as the monkey is reaching in a particular direction, in this case to the left, the neuron fires more strongly. So these are rasters with each individual dot indicating a neuronal firing. This is the go cue, and as the monkey moves to the left, this neuron fires more, and as they move to the right, this fires less. And this is a property called cosine tuning. So we've taken good advantage of this by looking at the firing pattern across an entire array. So you might expect, these are very small. They're 3.2 to 4 millimeters square. And if you think about the somatotopic organization of the brain, you would think, well, these are probably all doing about the same thing. Well, it turns out that this is actually not the case, that each of these neurons, they may be adjacent to each other, they're tuned to different reach directions. And so we can leverage this. And so if we denote each one of these, each firing rate by an arrow, and the size of the arrow indicates firing rate. If we ask our participant, monkey or human, to move up and to the left, those neurons that indicate up and to the leftward movement will increase their firing. Those that indicate the opposite direction will decrease their firing. And we can decode with a fair degree of accuracy the firing patterns across that entire array, allowing us to move a cursor on the screen. So here what you can see, and unfortunately the sound isn't working too well, but it's not necessarily that important here. This is a woman with ALS, who was one of our research participants, who is moving this cursor by imagining movements of her right hand. We're actually asking her to actively suppress movement and just imagine doing it. And she's moving this cursor around an optimized keyboard and answering the question, How did you persuade your sons to practice music? She's a musician, a luthier before she got AL. She made violins and violas and things. I mean, she's just an amazing woman. And she's just in a free-form fashion typing out the answer to the question here. And across the bottom, what you can see is an instantaneous rate of information transfer in correct characters per minute. So she's, throughout most of this averaging, she's hitting peaks of about 40 correct characters per minute, which is about 8 words per minute. So this is something that my laboratory has been interested in ever since I've been at Stanford, ever since I started working with Krishna Shenoy, my neuroscientist engineer colleague. And this is a very nice figure on communication brain-computer interfaces published in JAMA by Eddie Chang and Gopala Anamachpalli in 2019, indicating the speed of the various different communication modalities that we can employ. So if you go sort of over here to the left, you get these sort of really crude sip and puff interfaces that people can use to run wheelchairs. You have BCI-driven cursors, and that's kind of where we were with this 2017 paper. And then these increasingly high-performance methods of communication, all the way up to conversational speech, which is 150 words per minute, unless you're a New Yorker, when it's about 220. And so our goal here was to try to continue to increase our communication rate. So starting off with this 2D cursor BCI, which was directly translated from non-human primates. So this is exactly the same thing we were doing in monkeys. We brought it to humans. So this was sort of proof of concept that we could do it. So the next thing was to say, are there other ways that we might leverage in order to improve communication rates? And it turns out that if we focus on something like handwriting, we might be able to do that. And so the idea behind this is that you've got these arrays implanted in the brain, you've got your computer system, you place a prompt on the screen, you ask the person to imagine handwriting, and then you decode those higher dimensional movements from the cortex. So, I mean, the question right off the bat was, are these representations even existent? I mean, are there still these, very precise representations of handwriting after years of paralysis? And here's where our arrays are, sort of in this hand region of the motor cortex. This is the task structure, placing a letter on a screen, asking the person to write the letter, putting another letter on, tediously having them write letter after letter, many different examples, having them write words, having them write sentences, and then taking all of that and looking to see if just doing a very simple velocity vector reconstruction using a linear decoding matrix, it turns out that we can reconstruct pen-tip trajectories from brain activity, which is, I think, pretty cool. These are recognizable letters that are being extracted from brain activity using a simple equation like this. And that's good, that's fine, but it's not really the sort of richness that we need in order to do high accuracy decoding. So how would we accomplish that? Well, the way we did this was, and this is Frank Willett, one of the research scientists in my lab, who is going to be an assistant professor, joining me in early next year, which I'm very excited about. So we take all of these features, these threshold crossings of neurons recorded from these arrays. So this is demonstrating that electrode numbers all the way up to 192, which is we had 2 arrays, each with 96 channels. That's 192 channels. We take all of those different features, we throw them into a recurrent neural network, the details of which I'm happy to discuss if anyone's interested. And this will then output both transition probabilities, meaning that we're going from one character to another, as well as the probabilities of any particular character being generated. And some of these, as you can see, have a higher confidence than others. N is one particularly where, this could be mistaken for an H, right? So the raw output here of this particular RNN was TNE paper, which we know is probably not an English, are probably not English words. And so in order to correct this, we use different types of language models. And this is happens to be a bigram language model that looking at the probabilities of any character being typed given the previously typed character. And so then we can go through this search procedure and then autocorrect this. This is sort of like a sophisticated autocorrect. So using a system like this, we were able to achieve a marked increase in our characters per minute. So as you recall, our previous participant was at 8 words per minute with the point and click interface. And so this is participant T5, who is now approaching 90 words, 90 characters per minute, which is about 15 words per minute. So double the communication rate. And then by using a language model, the raw error rates can vary from day to day, from anywhere from single digits up to fairly large errors. But with the language model, we can correct that to single digits or on many days close to 0. And so this is what that looks like. I'll first show you at the bottom T5 using that same interface that I was showing with our participant with ALS. T5 has a spinal cord injury, and so he's imagining movement of his, he actually imagines a puck that he's moving across a table in order to move this cursor and copy the sentence, imagine all the people living life at peace. And then this is the real-time counter showing how long this takes him. So I'm going to rerun this video again, and this is about the same typing rate that, that T6 was able to achieve, Our Lady with ALS. And then at the top, what you'll see is running concurrently is using the handwriting decoder. And so as you'll be able to see, and he's just writing each paper, and again, he has this visualization of writing with a pencil on a yellow legal pad, and this is his very vivid imagination of how this goes. And this shows what that looks like. And so it's not perfect, of course. There are some occasional errors. And part of the reason that that this takes longer is that there is the opportunity to correct letters, whereas here there is no opportunity. But one could come back, obviously, with a language model, a more sophisticated language model later on and correct this. So this is, again, about double the communication rate. And then so just to recap, about somewhere in the neighborhood of 90 characters per minute with accuracies in the 90s. So this was something that we got pretty excited about because it was the kind of the next step function in communication. Of course, you can also use this in a free-form way. So here he is answering a few questions for us. What is your favorite meal for dinner? As you can tell, he doesn't live a very healthy lifestyle. He likes mainly fried food. We ask him what he likes to watch on TV. So he likes TCM, so he mainly likes to watch old movies. But he's able to use this interface in a free-form fashion. So I don't know how many people are familiar with this book and this movie. This is the story of Jean-Dominique Bauby, who was the editor of Elle magazine, and suddenly had a brainstem stroke. So he was left with the ability to only move one eye. And he wrote this entire book by having an assistant scan across letters, and then when she reached the letter that he wanted to indicate, he would move his eye. And so I recommend, if you haven't read this book, this is mandatory reading for everybody in my lab. It's, as you might imagine, it's a short read, but it's also extremely poignant and really, really illustrates the quality of life for someone. He would expect that a quality of life for somebody like that would be terrible, right? Because they can't do anything. Well, it turns out that's not the case. That, even people who are incredibly disabled have a lot to live for, and giving them the ability to communicate again is really, I think, one of our highest goals, one of my highest goals. Okay, so we've gone from cursor control to handwriting, but as we saw on that graph, there's still a lot of headroom. There are a lot of the we're at 15 words per minute, and people communicate at 10 times that rate normally in active conversation. So studying speech is, of course, there's a huge translational need for people who can't speak, but it's also really an exquisite motor control problem. I love this MRI scan. It's unbelievable, the coordination of all of these different articulators and all the different muscles and joints and things that and most of these, of course, aren't joints. They're just soft tissues that we're using to communicate at these rapid rates. And so understanding how this works is really tantalizing for the neuroscientist in me. So in order to sort of start to make headway on this, we, several years ago, this is our participant T5, who you just saw using the handwriting interface. We asked kind of a crazy question. We said, okay, we've got these two arrays, 96 channel arrays, firmly in the hand area of the cortex, the Ussery's knob region. What if we just tried to decode speech from there? It seems ridiculous, right? Speech isn't going to be represented in the hand area, but it'll just take a session. Why don't we give it a try? So, we designed this experiment where we, had him just say a few simple syllables and words over and over again, enough times that we could, get a representative sample of both his vocalizations and his neural signals. And this is, again, just to kind of show where his arrays are located. So this is the hand area right here. Central sulcus is here. And so this is in the hand region. And then, this was published back in 2019 in eLife. Sergei Stavisky, who's now at UC Davis, doing terrific work there. And to our great surprise, if you look at these raw rasters, here's the go cue, where we ask him to begin speaking these individual phonemes and syllables. What you'll see is that for each raster, there's pretty evident increase in neural activity and a decrease just before voice onset time with a concurrent increase. So this demonstrates that there are individual channels within the hand area, the motor cortex, that are modulating to speech. And if we look at any individual channel, we can see that we can differentiate. There are different firing rates. So this is in instantaneous spikes per second, that different phonemes and syllables and words are represented differentially. Like, for example, this neuron really likes k. Likes other things too, as compared to the do-nothing condition, but really likes ka. So this was really encouraging to us. We did a little bit more investigation, looking at various different mouth movements and discovering that, interestingly, some of these electrodes responded only to syllables, some only to orofacial movements, but most of them responded to both. So there are different representations that have to do with speech as unique from orofacial movements, which again was an interesting and sort of surprising finding. And then finally, we were able to decode with a fair degree of accuracy, about 85%, just using a very simple multi-class support vector machine, single classifier, to do that. So, very interestingly, of course, here we are in the arm-hand cortex, and we're showing that even though the modulation is not huge, there is modulation to movement of the mouth, face, and tongue, as well as what we would expect, modulation in the upper extremity, but also in other body parts. So the head, the leg, the eyes, the tongue, and even higher dimensional cursor control. So what this told us is that the brain is weird and interesting, which, of course we knew, but we never really expected this level of intermixing. And so we asked, well, what could this be? Is this cortical remapping due to tetraplegia? No, probably not, because you would expect that you wouldn't, as we saw, there's the majority of the modulation is still with upper extremity movement. So you would expect that to be gone and for everything to be, really remapped to orofacial movement rather than upper extremity movement. But we think this is the case, and I'll show you some more evidence for this. We just put this out as a preprint, so I'm excited. Fingers crossed, it's in review now. Hopefully it gets accepted And this is really exciting, this intermixed overlapping representation within the precentral gyrus. But most importantly, I think, is that this gave us proof of principle for a speech neuroprosthesis. So this very much de-risked this idea of placing arrays, penetrating arrays that go into the cortex into speech areas of the brain. And that's something that we wanted to have some fair amount of confidence that we were going to be able to achieve something with it before we did that. Because, even though we have, find some wood to knock on, never seen any deficits from any of the implants in the brain gate study, which we're now up to 17, there is a theoretical risk whenever you put something into the brain that you could cause some problems. Okay, and so that led us to T12. So this is participant T12, in whom we placed electrode arrays in the ventral precentral gyrus. So this is the orofacial area, as well as inferior frontal gyrus, which is canonical Broca's area. And this was based on some fundamental studies out of UCSF. So this is Eddie Chang's group, demonstrating that with using ECOG arrays, you can show that there are these different patterns. So this is high gamma power in the various different electrodes along the central sulcus. And what you can see is these different syllables are represented differentially in terms of where that high gamma power is represented throughout the cortex. And so we looked at this region and said, well, I think someplace here is probably not a bad idea, and probably right there is probably good. We actually went one further and used some imaging from the Human Connectome Project. We've been relying quite a bit on this data set, which it's a multimodal parcellation of the human cortex based on a number of different modalities, including myelination, connectivity, resting state fMRI, task fMRI, et cetera, to help us find our location. And so here, this is me using the stealth to localize these areas. And here you can see central sulcus here, which is ending right there. Precentral sulcus, this is the inferior frontal gyrus. And then Sylvian Fisher's over here someplace. So this is medial, this is lateral. And as you can see, unfortunately, there's these big vessels right here. This is about the size that the array is. And so we had to move that array a little bit north. So this one up here, this one down here, and then the two in IFG. So if we look at these inferior frontal gyrus, or I'm sorry, the precentral gyrus electrodes, we're very fortunate to get outstanding firing on almost all channels. So we can then, as before, take those firing rates from each channel, or even multiple neurons for each channel, and look at how they're tuned. And so we asked our participant to do a number of tasks, like, for example, forehead movement. So, wrinkle up your forehead. And so if we just pick a random channel here, this is channel 46, channel 47, and we look at the firing rate, or the fractional variance accounted for by that individual channel, this particular channel really likes forehead movement. So, and we denote this by a darker color. And so this is array number 1, this is array number 2. Each one of these dots is an individual channel. So if we look at the various different types of movement that we asked our participants to do, what you can see is immediately this pattern sort of emerges, in that these individual movements of orofacial musculature are pretty highly represented in this more dorsal array. And the pattern is different depending on the muscle, or the muscle group that's being moved, with a sparser representation on the ventral array. But if you look at phonemes and words, this representation is flipped. So there's actually a specialization. There's an area here at the most ventral part of the precentral gyrus that's speech specific, and which we've now started dubbing this the speech hotspot. And this shows up on the Human Connectome Project data. And we've now redemonstrated this in 4 participants total. So this is a real phenomenon. And I'll show you some more data on this later on in the talk. So again, same diagram as I showed you before for the handwriting decoder. Instead of putting the prompt on the screen. We can speak it or put it on the screen, either one. Our participant attempts to speak, and then their decoded speech appears on the screen. And using the same decoding method. So taking all of these features, including threshold crossings and spike band power. We don't do spike sorting. We found that there's very little benefit to sorting individual spikes. We get almost exactly the same information content from throwing all the threshold crossings into the decoder. And spike band power does actually give us a little bit extra performance as well. This is the very high gamma activity that probably represents spiking activity in an LFP sort of way. So this should be familiar from the other slide. We use a multi-layer GRU, recurrent neural network, which then, similar to what we saw with handwriting, spits out phoneme probabilities and transition probabilities. We can then put that through a language model as we did before. And as we've continued to advance these techniques over the years, language models have gotten better and better. So this publication late last year was using a trigram model. So we're now using more sophisticated models like GPT, which can give even obviously better accuracy given the inputs. Okay, so this is how this looks. And unfortunately, we couldn't get the sound to work, but maybe this will work. So this is participant T12. And so we've put a prompt on a screen, and this is for her to copy. So she's to speak this. She has bulbar ALS, very slowly progressive. She's still able to get herself around. She can still type on a tablet keypad, but she has a very difficult time speaking. So she's extremely dysarthric, as you'll hear. So we're having her attempt to speak this phrase, and then the decoded output appears below. So this is what it sounds like.
I don't want to call her a babysitter.
Then we have a speech synthesizer.
That would be good. I did well in school.
So to be fair, this video is a little bit cherry-picked because she's on a roll here. It's not always this accurate, particularly at the beginning of a session. It improves in accuracy as the day goes on. And similar to the handwriting interface, she can use this device in a free-form way as well. So we ask her here, what are you proud of?
I have a very good friend and sister.
And interestingly, and this is another avenue that we're pursuing pretty actively, she doesn't have to speak. She doesn't have to phonate. Here, we're asking her to simply mouth the words. And in fact, we can even decode inner speech, so without her having any overt behavior whatsoever. But as one might expect, the less overt the behavior, the lower the decoding accuracy. And so that...
I do not have much to compare it to.
For those who might be going to Society for Neuroscience, Aaron Kunz, one of the grad students in my lab, will be presenting that work, and we should have a preprint out on bioRxiv on this
I don't read as much as I
pretty
want
soon.
to either.
Okay, so at the time when this was published, and the field is moving very quickly, things are now better than they were even at the end of 2023. But this is the results. Still what I would call an unacceptably high word error rate. So 25%, approximately 24%, that's too high for you to be able to communicate in a reliable way. Now, if you're talking with your family and they can probably kind of figure out what it was you meant to say, this is probably okay. But if you're having a technical conversation of some kind, this is obviously not sufficient. But the communication rate is, so if you remember our sort of march upward from 8 words per minute to 15 words per minute, and now 65 words per minute, we're definitely increasing the rate of message generation by quite a lot. And this is on a general 130,000 word vocabulary. The previous published speech synthesis was using only a 50-word vocabulary, which is useful. You know, you can do a lot with 50 words if you need to express basic fundamental needs, for example, or medical things. But you really want a general vocabulary in order to be able to generate any message for anybody at any time. And so just as a comparison to the prior work, we did use the same 50-word vocabulary, about the same words per minute, and with a markedly improved word error rate. Okay, so we're still working our way up the spectrum here. So here we are at 65 words per minute. The question is, can we get to this point with people who have dysarthria, like for example with ALS? And interestingly, this is actually something one of our students is looking at, is this trade-off between speed and accuracy. If you go faster, are you less accurate? Are you more accurate, slower? Or is this just a preference of people who have adapted their communication style to their disease? And what I'll say is that there's work which I think I have a video at the very, very end, if we have time, which I can show, from our colleagues at the University of California, Davis. They recently published, and this has only been about a month ago, published a paper in New England Journal of Medicine demonstrating full independent use of a speech decoding system. Their participant uses this 12 hours a day. He has ALS and has really lost the ability to communicate with family and friends for the most part. So he's back to work now as a environmental activist and using this speech synthesis system on, as I said, 12 hours a day to communicate with his family, to communicate with friends and with colleagues at work. So this stuff works. I think we're well on the way to providing real benefit for people with communication disorders. Okay, in the last 15 minutes or so, I'd like to, now I want to leave some room for, I think we'll have time for questions, hopefully. I want to switch gears a little and talk about fundamental neuroscience. So part of the reason we do this, obviously, is to develop assistive technologies for people with disabilities. That's number one, right? That has to be first. But as part of that, we also, and this is just a privilege that we have as neurosurgeons, and particularly in the functional side, if you think of stereo EEG or ECOG or the early experiments of Wilder-Penfield, we have access to the brain. We have the ability to look at how the thing works. We've got the hood open. We can take a look. And so to me, this is equally exciting because we've got arrays in areas of the brain that have never been really investigated at this single neural level before, at the neuronal ensemble level before, using behaviors that really can only be experimented on, or that can only be investigated in people. And non-human primates, for example, don't talk. Only people do that. Okay, so I showed you this. This is that graph that I showed you before. This is from our paper in 2020, looking at a compositional code for various different limbs represented in the dorsal part of the precentral gyrus. And just to reiterate, we think that this classical representation is incomplete. The homunculus, really, it's a useful construct in some ways, and yes, indeed, at a gross level, if you stimulate the cortical surface, you do have dominant areas of the brain that are represented, But the single neuron somatotopy at the level of neural ensembles is more distributed and overlapping than what we see with cruder methods like electrocorticography or electrical microstimulation. So, looking at the dorsal part, the hand area, we see representations of everything. And in fact, looking at the ventral part, so down here in the in the face region, we also see representation of almost everything throughout the body. So this seems to be a generalized principle of the precentral gyrus of the motor system, that everything is everywhere in a way. So we investigated this in this incredible work by Daryl Deo, who was a postdoc. He was a student in my lab and then a postdoc in my lab, and now I'm proud to say he's working with Synchron. So he's he's gone off to industry where he's doing a lot of good work. But this was something that he really championed, was this idea of a different way to think about the precentral gyrus. So really, instead of thinking of it in a homuncular way, thinking of it as three separate, distinct pieces. There's a dorsal precentral gyrus, there's a middle precentral gyrus, and a ventral precentral gyrus, and each one of them seem to have different characteristics. And this was an incredible piece of work because each one of these letters over here is a research participant. So he assembled 1, 2, 3, 4, 5, 6, 7, 8 research participants across the BrainGate trial as well as University of Chicago and Pittsburgh. So he's assembled all of this data, and this shows the number of arrays. So for example, T17 has 2 arrays in the dorsal precentral gyrus, 2 in the ventral. T15 has 1 and 3, T12 has 2 in ventral, et cetera, et cetera. And so by aggregating all this data together, we can begin to get a real representation of inter-individual variability and how this, and whether or not there's a generalizable principle here. Of course, the other part of this is having each one of those participants do the exact same task. So this is, again, just an amazing amount of work. It's a simple task where we have them do just we call it the whole body sweep. So we just have them do a series of whole body movement tasks. And this is an example. We prepare it, go, return, and just keep doing that over and over again. It's pretty boring, but thankfully it doesn't take a whole lot of time for them to do this. And what we see is sort of as you might expect from the data that I showed before, where in the dorsal part of the precentral gyrus, there's hand representations, not unexpectedly. In the mid precentral gyrus, there's speech. This has been described by Eddie Chang pretty extensively, that there is this sort of speech region in the mid precentral gyrus. And in the ventral precentral gyrus, also tongue, speaking, tongue movement, et cetera. So these are all pretty normal and pretty expected, that there would be differences in the dominant types of activity that one might see in those regions. So this is going to be, this is a big figure. I'm going to try to break this down piece by piece. So let's just look at this participant. One array, so one, and I believe this is a 64-channel array in C2D2. So D2 means the second array in the dorsal precentral gyrus. This is participant C2. And then this is the whole body sweep. So these are all the different tasks that we ask our participant to do. And in this dorsal region, what you see is that, and this is normalized modulation. The brighter colors are more modulation, and 0 is down here. And what you see is that, as expected, you get a lot of modulation to arm movement, lesser modulation to contralateral or movement of the ipsilateral arm to the side of the array, which again is not unexpected. Some modulation to leg movement, some to face, some to head, and only this X. So this is the only condition in which this array has no significant difference in tuning. So if we look at everything across everyone, so this is all of those arrays with all of those conditions across all of those participants, patterns begin to emerge. And what you can see is that, in these dorsal precentral gyrus arrays, Again, not unexpectedly, you have arm representation, but you also have a lot of leg, contralateral arm, contralateral leg, face, head, and even some speech. And again, and these are arranged roughly from dorsal to ventral. So sort of the closer that you get to this mid-precentral gyrus, the more kind of, I guess, speechy things get, the more sort of speech tuning you see. And in the mid-precentral gyrus, there's very strong tuning to speech, lesser tuning to face and head, but really broad tuning. So arm, left arm, left leg, right leg. And similarly, in the more superior part of the ventral part of the precentral gyrus, so that's below that mid-PCG, you also get this pattern of broad tuning. And then as you get more ventral into what we call the speech hotspot, you get very, very much speech and less of all these other things, but still significant tuning. So only a few channels are not significantly tuned. And then you can look at this in a different way. Each of these is an array, and this is just the sort of the aggregate activity in that entire 8x8 or 10x10 array, showing the same sort of pattern. This is the inferior frontal sulcus, superior frontal sulcus, and these are all those locations along the precentral gyrus, showing that indeed there's a gradient of tuning, but also tuning to many other different conditions. So clearly more complicated than what than what some of the textbooks say. Okay, I'd like to just finish up my last few slides talking about some other interesting things that we've been doing. So this is decoding multiple simultaneous finger movements. So this is participant T5. Doing this really interestingly difficult task. I've tried doing some of the tasks that he's able to do, and I can't do them. So again, he can't move his hand, but this is the decoded finger positions in real time as he is, moving them, to various different positions in order to encode different gestures. So this is really rather high accuracy. This was Matt Wilsey, who from the University of Michigan did his fellowship with me last year. And this now is actually, I'm happy to say, accepted at Nature Medicine. So this should be published before too much longer, demonstrating this extremely accurate decoding of finger position. And we took this and did something fun with it. Our participant T5, who has been in the trial for a long, long time, from the very first time I talked to him, he said, I want to fly a drone. I want to get out and fly this thing around my neighborhood. And so what we've done is, although it would be prohibitively difficult probably to actually do this in real life, we'd need a gymnasium and a drone and everything. So, and by the way, we have had him flying a physical drone in our drone lab in a controlled environment remotely. So he's been able to do that. But this is just demonstrating the degree of control he has using these different axes of finger movement. And he's actually really good at this. He's better. I don't know how many of you have tried to fly a quadcopter, or at least the kind that don't have a lot of assistance, and it's not trivial. And so he's controlling all the different axes on this quadcopter and making it go exactly where he wants it to go. So for him, this was sort of a fun thing that he could do that really fulfilled one of his major goals in the trial. Okay. So, with that, I think I'll wrap up. I hope that I've shown you that the field is moving fast. We're moving closer to clinical reality. To me, not only is that important for restoring function for people with disabilities, but also the fundamental human neuroscience insights that we can discover. And we're discovering more and more day by day. It's not going to be very long, and we're going to have real systems that can provide assistance for people. In fact, the day is really here, with the Synchron trial providing in-home assistance, with the work that they're doing at UC Davis, which is providing real assistance. So the future is almost here, and it's really, really exciting. These are the people who actually did the work. This is my incredible lab. Chewy, of course, is an integral part. Chewy is the only dog who made it here. There's a couple others that didn't, unfortunately, make it into the picture, but Chewy is one of our lab mascots. And my dear departed partner, Krishna Shenoy, one of a real giant in the field of neuroscience who passed away in 2023, unfortunately in 2023. And we're coming up on the 2nd year anniversary of his death. Of course, we're funded by a number of different institutes, NIH, some private funders, and then of course our participants are absolutely incredible folks. To find people who want to participate in this kind of research is a rare thing because they know that they're not going to necessarily get personal benefit, but they're going to be pushing the field forward for those who follow. And we're part of a larger consortium called BrainGate. So this is a consortium that spans 5 institutions, 2 coasts, including the middle of the country, and just an incredible group of people. This is at one of our annual gatherings out at Half Moon Bay, actually, which is really a nice place. So thanks, and with that, I think we've got about 5 minutes for questions.
Very exciting, beyond exciting. Questions, comments?
Jay?
Just a quick question. When the patients are controlling the cursor, you describe them thinking about they're having a knowledge, for example, she's thinking about where her hand puts nature.
Yeah.
Is there a point in which patients are transitioning where they're just thinking about moving their cursor and not having to supplement a different body? I'm interested in that experience.
Yeah, that's a great question. Yes, when T5 is in the flow, he said things are just moving. This was, once he got good at controlling the drone, things were like that. You notice the display, we have an indication of his finger positions, which he had to use early on to learn the mappings. But later on, he rarely paid attention to that screen. He just felt, he just controlled the drone in an intuitive way. We also had him doing a 5D cursor task. So moving a cursor in 3 dimensions and then matching its orientation in 2 different rotational degrees of freedom. And he couldn't describe what he was doing. He said, I don't know what's happening, but I'm just making it do that. And so, we wonder if with enough practice and with enough exposure, if maybe new representations are actually being formed. This is something that it's a really interesting question, is this idea of learning in BCIs. We've barely investigated it. Other labs investigated a lot. We really haven't done that, but I think it's a hugely interesting area of investigation.
I remember seeing a talk a number of years ago that the gentleman was, you may know, I can't remember his name right now. I like a vest that gave contact.
Oh, yeah.
Eventually, the person could interpret that as different information on because they wouldn't even think about it. And I think that sort of plasticity of the brain, think about they play video games and they no longer think press the A button, press the X button. They shoot or jump or whatever it needs.
Right.
And so I think it's interesting how the brain can wire that way. When you look at revolutions it's done. There's a part of not just reading the brain, but rewriting the brain.
Yeah. Mm-hmm.
Thank you. It's just such an inspiring talk, and I think the positive applications are clear. We're all wires or cell phones, as you can clearly see. I would love your opinion about a little bit of the sinister aspect of this. When you showed the drone flying around, it gives me a little bit of shivers as to just the evolutionary biological advantage this is going to give to some humans over the next 20 years. What can we do as a neurosurgical community to sort of stay in control, or at least to have that morality stay?
Yeah, so it's a great question. I think ethics is a huge part of this. Even as late as 5 years ago, I would have said, we don't have to worry about this stuff, ethics, it'll come, we're 50 years away from that. Things are moving really fast. And a lot of that, I think, has to do with the amount of investment that's being poured into the field. So we have to worry about these things. We have to be concerned about ethics. I think we can't leave the ethics to the ethicists, which sounds kind of weird, but many of them are not very practical people. They're philosophers who, think about things in a way that's theoretical and not practical. So it's really important that we take an active role in defining some of these ethical principles about when devices should or should not be used. And it's tough because, we're neurosurgeons, we're pretty practical. We tend not to think, at least I'm speaking for myself, I think very mechanically. But having, some of those collaborations and people to work with and choosing practical ethicists. And I'll throw out an example. Joe Finns at Cornell. Joe Finns he's actually an MD, but he's no longer practices medicine, he practices ethics. But he does it from a very practical standpoint with guidelines and outlines and not just pontificating. And so I think that's what we need to do, is have clinicians on the ground, neurologists, neurosurgeons, people who will be working with these patients, picking the people that they want to help move the field forward, who can do it in a thoughtful and practical way without coming up with these weird hypotheticals that could really torpedo things in a way that is actually unethical, in my opinion. I think some ethicists are not terribly ethical, Ben.
No, I feel like I read what you write. I think it's you and the audience. Just great to hear you. Great to hear you present the work that.
Thank you.
Thanks for coming here. You mentioned, second part of the talk, that many of the participants in the trials do so in a kind of altruistic way, which isn't help raising gift that they give to the field. But as we move towards these kinds that are really meant to help restore function, you have to think practically about clinical trials. I know a number of us here were at that FDA workshop last week. What's your thinking on actually how to measure and quantify the benefit that these interfaces are providing to patients? It seems so intuitive when you see that what you're providing is something very meaningful. And yet when you to your point on practical ethics and practical things, when you set about trying to measure and quantify the benefit, it turns out to be nuanced. What's your thinking about that?
Yeah, no, thanks for putting me on the spot. Given that we were just at a workshop where something like 100 people were debating this very topic, more or less productively, I would say. So I think we're at an early stage. What are the clinical outcome assessments that we need to have to demonstrate efficacy? And those are tough. I think, and many of these discussions really featured a couple of different axes, one of them being technical and the other being, more, I guess softer in some ways, more quality of life types of things. I think both of those are necessary. I think we have to focus on technical factors. How well does the thing work? What are your words per minute? What are your, how many neurons? I think those things are important as underpinnings. But I think in order to really demonstrate benefit, we have to have, and we probably have to invent new metrics, really. I mean, we can borrow from the literature, but, and there is some good literature, particularly, in my area of communication, the AAC literature, Melanie Fried-Oken, who's, been very active in developing these types of things. So I think we can borrow things, but we also have to really carefully think through how we want them to be represented. So I know it's kind of a vague and smushy answer, not maybe exactly what you're looking for, but I think what it demonstrates is we still a lot of thinking to do, and a lot of investigating what's the real capability. Again, just over the past couple of years, we have capabilities I wouldn't have dreamed of a couple of years ago. When we implanted our participant T12, nobody had put microelectrode arrays in the ventral precentral gyrus. Now we have 4 patients, and they're using these systems 12 hours a day, which is really, I mean, I couldn't be more happy. It's incredible. Yeah, amazing times.
Thank you for the awesome talk, Dr. Henderson. My question is, how well do you find proficiency in certain tasks translate to other tasks? For example, like the drone flying game. If you were to switch that to like a car racetrack versus like a patient actually driving a car, how do you find they progress in terms of proficiency?
Yeah, I mean, I think there is sort of a baseline proficiency in just using a BCI. You know, kind of being aware of the parameters of, you know, what you're doing with your brain and how it's going to affect the world. T5 unfortunately passed away last year after being in the trial for 7 years, going on 8. So over that 7 years, he kind of got a pretty good idea, you know, how does a BCI work? But, you know, each task is individual. Being good at one thing does not make you good at another necessarily. And so as we designed those various different types of research sessions and different tasks for him to perform, sometimes he had to get practiced at it. And he took it very seriously. Amazing guy. He was like, for example, with the optimized keyboard, he asked that we print it out so he could put it on the ceiling above his bed so that he could, you know, glance up and just memorize it, because he wanted to memorize the keys so he could go as fast as possible. You know, he said, I'll be your fastest. You put me up against those monkeys any day, I'll kick their ass. Competitive guy.
To that point of, you know, you're in some of these patients, you're dealing with a very progressive disease. Are you seeing differences in how you are measuring or dealing with the metrics and changing as things progress?
Yeah.
Does that alter the recording or the assessment of the metrics?
Yeah, that's a great question. It's and it's tough to, in a way, it's tough to assess because there are a number of factors that go into array performance degradation. And that is one of the challenges with this technology, is that over time, you can sometimes see a roll-off in performance. We think that that mainly has to do with physical features, changes in the electrodes themselves, you know, changes in the local milieu, scarring, all sorts of different things. But, you know, there also could be biological factors that are involved in the disease progression. We've been fortunate that most of our participants have been pretty slowly progressive ALS. I don't know that we've got anybody who's rapidly progressive, but it will be interesting to try to study that if we have that opportunity. And, you know, if you get to the point where someone's completely locked in, what signals are available? Yeah. Does the brain just become disconnected and they can't communicate anymore? So these are things that we now have the capability, I think, to study. Having so much data now and what to expect in most cases, we can now approach those outliers with a little bit more confidence.
Well, I'm sure there are going to be a lot more questions during the rest of the morning, but we get a chance to spend more time with Dr. Henderson. Thank you again, Jaimie, for coming.
Thank you. Thanks for the invitations. Terrific honor. Thanks.
Thank you.